is Tarek Shazli. I'm a faculty member here in the Mechanical Engineering Department at the University of South Carolina, but also part of the Biomedical Engineering program. So the Biomedical Engineering program is split between three main departments. It's Mechanical Engineering, Chemical Engineering, and the Cell Biology and Anatomy at the School of Medicine. There's a total of approximately 30 faculty. They're evenly split between the School of Medicine and the College of Engineering and Computing. On the engineering side, we're more leaned towards the chemical engineering side. There's only a few mechanical engineers that are a part of the program. Our graduate student population is approximately 30, but that consists of 3 different degree programs. We have a Master's of Engineering in Biomedical Engineering, which is a 1-year coursework only program. It's 30 hours of coursework completed within 2 academic semesters. And we have currently 5 students that are in that program right now. We have a handful of Master's of Science students, so it's typically 2-year programs that have both coursework and research component to it. And the majority of our grad students are PhD students. We have approximately 15 spread out throughout our program. So areas of research include tissue engineering and immunomodulation. We have a large focus on biomaterials and drug delivery. Faculty at both in engineering and the School of Medicine focused on biomechanics. Drug development is a major area. Computational biology and computational medicine is an area of focus, and bioinstrumentation. The collaborative nature being across colleges is probably more than most degree programs. We have a number of graduate students who have in fact been co-advised by a faculty member in engineering and School of Medicine. It kind of adds an extra dynamic to their projects, gives them clinical exposure, and see the translational pathway embodied by many of their projects. The first lab we visited was the Gower lab. He calls it a lab for immunomodulation and tissue engineering. Within that lab, we saw some standard equipment like a laminar flow hood, fluorescent scope, a centrifuge, and a lyophilizer. A lot of his work is focused on using the immune system in order to direct the phenotype of cells for various clinical scenarios. He's currently using adipose tissue for various clinical applications or preclinical studies. The second lab we went to was Shanglu. His equipment that we saw there included a culture hood, a deep freezer, a centrifuge, a rotary evaporator, and just a pretty extensive setup of fridges and freezers for tissue and cell storage. His aspects include drug delivery and instrumentation to some extent. The third lab we went to was the lab director, Dr. Silke Henrich. This lab is used for both undergraduate teaching activities as well as a community resource for graduate students, which is why it was included. The equipment we saw there was real-time PCR setup, and again, standard equipment such as an autoclave, plate reader, and a bioimaging system. Next lab we went to after that, the PI is Dr. Gyun Wang. His work is really focused on advancing imaging capabilities, both in terms of time and space. The equipment we took pictures of there includes just a setup of an optical bench with a number of different types of microscopes and systems for high-resolution imaging. And then the second piece of equipment we saw was associated with his nanoscope setup. Next, we visited my lab, in which we saw my work is involved in cardiac and cardiovascular biomechanics. We saw 2 standard pieces of equipment for ex vivo mechanical testing of soft tissues. So that included a high-fidelity uniaxial mechanical tester as well as an inflation extension tester for biaxial testing of tubular constructs. And the final lab that you'll see images of is the lab of Dr. Michael Sutton. So Dr. Sutton has been involved in the biomedical engineering program in a number of collaborative projects that span engineering in the School of Medicine. His lab and his group has pioneered digital image correlation over the past decades and high-rate loading analysis. He has a company that branched off of this research, started here at University of South Carolina, that provides DIC hardwares and softwares across the world. One of our strengths is the collaborative nature of the research. So a lot of these labs are open in the sense that the equipment is not, free-for-all available for everyone to use, but with an email or a quick call, you can typically gain access to a wide range of equipment. So that spans both tissue preparation and imaging, sample characterization, different methods for synthesizing materials, techniques and resources associated with animal studies, drug discovery, immunohistochemistry, and a wide variety of different lab resources are going to be available to our graduate student population.